Assalamu alaikum everyone, welcome back to BioCore. In the previous lecture, we learned about enzyme specificity and how they work using the lock and key hypothesis. Now, here an interesting question, that is, why do we get fever? Why does boiling destroy living cells? So all of these questions are connected to an important concept, that is effect of temperature or enzymes. So today we are going to explore the enzymes behave under different conditions and why this is so important for life. So let's begin. We are going to start the basic concept of effect of temperature on enzyme. Enzymes are protein, and like all the chemical reactions, like increasing temperature increases reaction rate, decreasing temperature slow down the reaction. This is the very general. The next point is, why does temperature increase reaction rate? Because inside the cell molecules are always moving. This movement is called kinetic energy, and when the temperature increases, molecules move faster. Collision between the enzymes and the substrate increases. More collision, more enzyme substrate complexes, and the reaction will be faster. This will be very good. Now we discuss the optimum temperature. However, this increase does not continue forever. There is a specific temperature at which the enzyme's reaction works best. This is known as the optimum temperature. For most human enzymes, optimum temperature ranges to 37 degrees Celsius. At this point, the activity is maximum and the rate of reaction is highest. So this is the best temperature for the enzyme to do work. Now we discuss what happens at high temperature if we exceed the temperature. If the reaction temperature is 5, if we 5 degrees Celsius, when we started the rate of the reaction, the kinetic energy increases and the reaction rate increases, but only up to 37. After 37, what will happen? This is the very important concept, that at that temperature above the optimum, the enzyme's activity decreases rapidly because enzymes are protein and high temperature affects their structure. So this is the graph that you can see. Here is the reaction rate, that if the reaction starts from 0, the reaction rate is increases. You can see the graph direction is upside. So it's mean that the reaction is increases, and at 37, after 37, the graph rate will be on the downside. The slope will turn downside. This means that the reaction rate start decreases. So this is the basic point, and this is the graph that's showing the effect of temperature on the rate of enzymes control reaction, that at when the temperature is below than 37, if we start to increase the temperature, the slope will turn the upside. It means that the rate of reaction increases. But if the slope is turned downward, this will means that the reaction rate slow down, and this is just because the denaturation that occurs after 37 degrees Celsius. Now here are the very important point, that is the what is the effects of denaturation. When it's come to the high temperature, this is the high temperature, what happens that the bonds in enzymes to break and the shape of active site is to change. The change is very permanent, and this permanent change is known as denaturation. Now what is the result of denaturation? Substrate cannot fit, no enzyme substrate complex is formed, and the reaction stops. So this is the results after denaturation, that the reactions rate will be stops. Even if temperature decreases later, the enzymes will not work again. Like if we exceed the temperature up to 40, the optimum temperature is 37, and up to 40 to 50 the enzyme denatured. And if we again bring the temperature about 36 or 37, but the change will be permanent, so there is no reaction. Okay, if we change the temperature later, the enzymes will be not work again. Now there is a very important and the real life example, that is about egg. Consider an egg. Okay, think about an egg. There is a raw egg, that is in liquid stage. When you cooked the egg, it is becoming the solid change. This change is permanent and cannot be reversed. Some happens to enzyme at the high temperature. So the same happens to enzyme at high temperature. This is the same effect that is about the enzymes. Like if we once boil a egg, we can't reverse it, we can't bring it in its original position. So the enzymes is like the same. If once they are denatured, they will never bring to the same state. There are the some important enzyme concepts, that is temperature increases reaction rate due to more collagen. Optimum temperature gives maximum activity. High temperature causes denaturation. Denaturation is irreversible. Enzyme loses its activity. Active sites shrink. And what is the low temperature effect? We will discuss the low temperature effect. Low temperature effect is like molecules move slowly. Fewer collagen occurs, so reaction is slow, but enzyme is not damaged. If we increase the temperature, enzymes will work again. So this is all about the lecture. Let's quickly summarize this. Increasing temperature increases reaction rate due to increased kinetic energy and collagen. Optimum temperature, maximum activity, that is 37 degrees Celsius. High temperature, denaturation, mean permanent loss of function. Low temperature, slow reaction but doesn't damage enzymes. If you found this lecture helpful, like the video, comment your answers and subscribe to BioCore for complete biology lectures. In the coming lecture, we will discuss effects of pH of enzymes. See you in the next lecture. Allah Hafiz and keep learning with BioCore.